do so. So just some quick key concepts here. So remember that cellular transformation involves gradual buildup of mutations that take many, many, many generations of cell division. And so that's why we say up until that point, the transforming cells are almost preoccupied with their own sorts of strange issues that are taking place inside of them as they're gaining mutations and losing control of the tumor suppressor genes that release the brakes that enable, at the same time, mutations in oncogenic pathways to then take over. So we said that was kind of like monologue for many years of the cells just doing their own thing. And that's why it takes many years for a developing tumor to really get to a certain size, because most of the cells as they divide are undergoing apoptosis. They are still doing the right thing, so to speak, as tumor suppressor genes like P53 properly put an end to their further division. But as they gain more and more mutations, you begin to get different types of mutations in different little subpopulations. Remember that the tumor originated from one cell that began to go down this pathway of transformation, but at a certain point, there's enough copies of that one cell through clonal expansion that you begin to see different individual cells gaining their own set of new mutations. And so that's why then the tumor becomes heterogeneous. You begin to get subpopulations of different cell clones in different parts of the tumor, depending upon whether those mutations give them a proliferative advantage. So we also said that as long as it's contained within, let's say, the basement membrane, limited by the blood supply that cannot really provide much more oxygen or nutrients to them, then the tumor remains in situ. And that could still be considered a benign tumor. It is not necessarily cancerous. But in order to break free of that in situ limitations, that's going to require the help of the surrounding cells. And in this case, we are talking about the manipulation of the stroma, so that now you have this back and forth dialogue taking place between the transformed cells and the supporting stroma or mesenchymal cells that ultimately then create sort of this very dysfunctional household. That was the analogy that we had of this teenager rebelling against their parents, but ultimately up until that point, still needing their parents in order to provide support. So we said that's kind of like dialogue. So today we're going to take it a step further and say, what if at a certain point the tumor cells are ready to break free? They've gained enough mutations, they've done things like they've managed to stumble upon how to turn on the wound healing response. And so now the stroma surrounding them is very reactive and confused. And in that kind of dysfunctional situation, then it creates an opportunity for the cells to break out. So we can imagine that as the teenager. Finally, the household has just gotten just so dysfunctional. The parents just don't know what to do. They're manipulated. And maybe even it gets to the point where the dad gets so angry that he kicks the son out of the house, and that's the turning point. And we have a teenage runaway now from home. So the rebelling teenager runs away from home, but the real question is he prepared for what's ahead? Meaning now that he is out of the familiar confines of the home, can he really deal with being a runaway out in the real world? And I remember reading some statistics about runaways, and it turns out that something like 9 out of 10 teenage runaways who are found by the police within a few days of running away from home, they actually find them in places like the shopping mall, in places like GameStop or in McDonald's, something like that. And it turns out that runaways oftentimes go to those types of places because they feel comfortable there, to hang out there. And so that's a very important kind of theme or concept to keep in mind as we talk about the tumorous epithelial cell now leaving the in situ tissue that the tumor was originally formed in and looking for a place that they feel, quote unquote, comfortable to metastasize. So here are some illustrations showing examples of metastatic lesions. So this first one here, this is a metastases of pancreatic cancer that has now ended up inside of a lymph node and is beginning to colonize there. Over here, we have breast cancer metastases that has also ended up metastasizing in a lymph node. So it is no coincidence that lymph nodes are a very common place for metastatic lesions to form. Again, because you think about it, remember in the previous lecture, we looked at a cartoon that was outlining or illustrating the capillaries, the blood supply that goes to your tissues, and how the lymph vessels, the sort of in the cartoon, they were green colored, are draped over the capillary beds throughout our tissues as a way to drain things like extra fluid, bits and pieces of dead cells, etc., away from our tissue and send that material through our lymph system to be cleaned up. So not surprisingly, metastatic tumor cells that have been shed by the original primary tumor oftentimes end up being drained away in the lymph vessels, and they end up in places like lymph nodes. Here on the bottom is an example of metastatic lesions in bone marrow. So all these sort of light reddish small little cells are the red blood cells that are maturing inside the bone marrow. And then these blue ones are actually metastatic cancer cells that are now beginning to grow and divide in the bone marrow. And as we will talk about later, certain tissues like bone marrow are also a very popular place for metastatic lesions to form, because the environment of the bone marrow is very conducive to stimulating the growth of new cells. After all, that's where all your red blood cells and other cells of your blood are coming from. So you can imagine there's lots of growth factors in here that make it a very fertile destination for metastatic cancers to form. So if you think about this, a primary tumor has to get a certain size before it can be detected when the patient goes to the clinic and has some sort of diagnostic tests done. So a primary tumor has to be about 10 to the 10 cells before it's large enough to be detected. In a previous lecture, we kind of looked at some examples of how large tumors have to be before they can be picked up by things like an X-ray or even physically palpating, the physician actually feeling the lump under the skin. However, primary tumors only account for about 10% of cancer deaths. The vast majority of deaths due to cancer are due to metastatic lesions. In other words, it's not the primary tumor that kills the majority of cancer patients. It is the metastatic tumors that are responsible for 90% of cancer deaths. There's very high variability in the nature of these metastatic tumors, and a lot of that simply depends on the primary tumor type. And so this is another theme that we talked about, this idea that because tumors can form in pretty much any of the tissues in your body, and the original cells that give rise to them might be very specialized for different functions, then it's not surprising then that certain cells from certain tissues might actually be more ready to metastasize and more flexible in their ability to metastasize in different tissues compared to others. So this brings us to this outline that we can call the invasion metastases cascade. So it is a series of 7 sort of steps or 7 events that take place as part of metastases. So starting with step 1 here is the original formation of the primary tumor in the in situ environment where that original transforming cell started going down this path, started going rogue like years and years and years ago, right, until it reaches this point where it's big enough. So this lecture, we're going to step through these 7 steps, and hopefully you can understand all of the things that must take place in order for a tumor cell, cancer cell, to really metastasize. So let's start off with step 1 here, which has to do with local invasion. The implication here is that the cells must first somehow break free of the basement membrane that is containing them. And as we studied previously, we said that the tumor cells, they stumble upon certain mutations that kind of trigger the wound healing response. For example, they start secreting growth factors like PDGF, platelet-derived growth factors, that recruit macrophages, immune cells, to come through the tissue and actually associate with the tumor. And the matrix metalloproteases, the MMP enzymes that are secreted by immune cells like macrophages, they remodel the extracellular matrix, doing things like digesting away the basement membrane. And so this releases a lot of the growth factors that were actually being held in the basement membrane, that was limiting the ability of the cancer cells to have access to these growth factors before. As a result, you end up now having this dialogue taking place between the cancer cells, or the pre-cancer cells, and the stroma, and it creates this, what we call reactive stroma. This is the stroma becoming very dysfunctional and confused and manipulated. So that reactive stroma then begins to encourage the invasion of the tumor cells. So here, the tumor cells are beginning to make their way out of the original epithelial site where they were, through the mesenchyma, or through the stroma, heading towards something like the closest capillary. Right? Do keep in mind also, here it's pictured that as part of this tumor being ready to metastasize, you can see it has attracted the growth, or it needs to attract the growth of a lot of blood supply. Right? And that's how then these tumor cells can keep growing, and that also makes it far easier for these tumor cells to actually have an opportunity to nudge up or make their way to one of these capillaries. So in order for these epithelial cells to be okay moving through the stroma, that means they have to change from their epithelial state into more of a mesenchymal state to allow them to mingle with stroma and make their way through it. One of the important changes that takes place has to do with certain surface proteins that are involved in allowing something like an epithelial cell to sense not only maybe that it's sitting on a basement membrane, but also other types of surface proteins that help it sense that other epithelial cells are nudged up against it. And that's part of it being happy and functioning normally. So many of these adhesion molecules function like these cadherins. So here we have an example in the skin of epithelial cells. These are keratinocytes that produce the keratin that becomes the hard protective outer layer of your skin. And among these epithelial cells, you have melanocytes that are producing the melanin that's needed to protect the keratinocytes when they sense that their genomes are being damaged by UV radiation. So the melanocytes very much normally behave and function as one of these epithelial cells, and it mingles within the epithelial cell layers so that it can associate with the keratinocytes. And these cells interact through E-cadherins. So that's what these little blue things are that enable them to work together properly. So when you get a high dose of UV radiation, it's not as though the melanocytes are any more susceptible to UV damage than the keratinocytes, because they all get zapped by UV. But remember, the point is the keratinocytes normally don't last very long. So even if they end up getting mutations, if the mutations are too bad and the genome's too badly damaged, they will just undergo apoptosis. And we said that's what sunburn is, right? The keratinocytes doing the right thing, killing themselves for the sake of saving you, your entire body, from cancer. But normally, right, even if they got mutations that might be transformative and they don't die from apoptosis, they would end up being shed anyhow. So we don't worry about them. But melanocytes are meant to stick around and constantly be in the epithelium. So it's the melanocytes then that can really harbor UV damage and build up a whole set of transformations that end up becoming cancerous. That's why a very important type of skin cancer is melanoma, right? So it involves the transformation to cancer of the melanocytes. So part of this then is the melanocyte stumbling upon mutations that switch off the E-cadherin protein on its surface and instead switch on a different type of cadherin, N-cadherin. So N-cadherin is the molecule that's used by mesenchymal cells or stromal cells to happily communicate with each other. So by switching off E-cadherin, the melanoma cell no longer cares about hanging out with the other epithelial cells. It now takes an interest in mingling with mesenchymal cells. So these may be fibroblasts that are certainly part of the mesenchyma, the stroma, or maybe endothelial cells from nearby capillaries. So this then encourages the melanoma cell to be able to migrate through the stroma. All right, so that results in localized invasion. Right? The cells now being able to move outside of its normal epithelial space. The next step then would be entry into the blood supply. And so we call this intravasation, right? Entering the vascular system. You can see here, once it makes its way into the blood supply, these cancer cells then will have the opportunity to interact with things like platelets, lymphocytes, and other types of blood components that may have just been passing by in the blood. So as part of step 2 here, the tumor cells enter the bloodstream in order to be able to kind of make their way from the surrounding tissue into the blood. It involves cooperation, again, of a very dysfunctional communication between 3 types of cells. The carcinoma cells themselves, with the help of macrophages and the endothelial cells, the actual cells that make the wall of the capillary, all working together in a very confused way to enable this to happen. Not surprisingly, the tumor cells are going to attract a lot of platelets because of the PDGF signals that they're giving off. But equally importantly, this is not a smooth and easy ride here. Once they enter the blood is actually a very hostile environment for cells that are not meant to be there. So let's talk about this. So first off, let's kind of visually appreciate what we mean by there has to be this dysfunctional triad of 3 different cell types working together in a confused way for the cells to enter the blood. So on the left here is an image of carcinoma cells. All of these pink cells are carcinoma cells, and these are actually stained with an antibody against this MINA protein. So down here you can see MINA is a protein that helps regulate the actions of actin fibers within the cell. And so the more actin remodeling that you have, the more motility the cell has. So overexpression of this MINA protein is often associated with invasive cancer cells that are moving around, that are mobile or motile. But here we've also in dots here specifically highlighted the areas in dots on this one here and in circles on the one on the right, where you can actually see the 3 different cell types working together. So of course you have the pink cancer cells here within these circles. These spaces in between that are dyed black here, the black dye actually is staining the endothelial cells or the wall of the capillaries. And I know this looks really weird. I've discussed this with you before, that when you take a cross-section of a capillary, it may not look like much. It looks like 2 endothelial cells that you happen to stain here and a little bit of space inside. In this case, the space is actually occupied by a macrophage that's coming through. It's a nice reminder that capillaries are very tiny and the walls are very thin, literally just endothelial cells that are kind of wrapped around them. So in this environment, the macrophages, again, are secreting enzymes that are digesting away the stromal tissue or the mesenchymal tissue. And the endothelial cells themselves have to be amenable or willing to kind of move out of the way to create a little gap where these pink carcinoma cells can then shove their way in to the blood. Here on the right, you see the same thing again, but this is a really nice reminder that if you take a cross-section of a very large tumor right, that is now ready to kind of break free and expand. By then, the tumor itself is crisscrossed with an extensive network of capillaries throughout, right? That's the whole reason this tumor was able to suddenly blow up in size and really start growing. So each of these circles here, and in fact, not even the circles, but any of these locations where you see the black stained capillary cells, that represents an opportunity for any of these pink cancer cells to enter the blood supply. All right, so hopefully when you look at this, you realize and imagine then how many of these cancer cells are actually entering the blood at this moment, just from this teeny tiny microscopic view here. So I said capillaries are very limiting spaces. A typical capillary is only about 10 microns in diameter. I put the micro here in red because I realized for some reason there was a typo in the PowerPoint slides that I posted where it actually said 10 millimeters, right? That's obviously wrong. They're 10 micrometers. A typical red blood cell itself is 7 micrometers. So that means a red blood cell flowing through a capillary, there's not much wiggle room. And that's why these red blood cells, you've probably seen these animations before, where the red blood cells almost have to go through the capillary in single file. It's that tight inside the tiniest of capillaries, right? But that's how it's designed, because it's only if you have the capillary so small that you can have the diffusion of oxygen out of it to supply the surrounding tissue. And again, because the distance that the oxygen can diffuse once it's out of the capillary is pretty small, right? That's why you need to have many, many capillaries throughout our tissues, which is something that I think most people don't realize. A cancer cell then is much larger. A typical cancer cell is about 20 microns. Right? So you have a 20 micron cancer cell trying to make its way through a 10 micrometer diameter capillary. Immediately you can tell this is not an easy kind of situation. So in this slide, to make it easier to visualize, you can see here, these would be the walls of the capillary in yellow, so the endothelial cells. You can't really tell which one are the endothelial cells, but this is the diameter here. Then you have a red blood cell that you're looking at it from the side, right? So instead of looking flat on, you see it round, you're looking at it from the side, so it's kind of concave like this. Here's a white blood cell. So you can see it's really cramped. I think these are some other red blood cells that are squeezing their way through there. It's not a very easy environment for the cancer cell to suddenly find itself in. In addition, once it's in the blood, cancer cells will tend to clump up. Why? Because cancer cells, because they're secreting a lot of platelet-derived growth factor, they will recruit things like platelets. And then these platelets begin to clot around the little clump of cancer cells. Again, think about this. This is the wound healing response, right? The tissue believes that this is a wound that needs to be patched up. So lots of platelets are going to come and forming a clot. So you'd see here, this is a typical clump of cancer cells. I believe these little reddish stained ones or smaller bits are actually fragments of platelets, although I'm not sure because I don't know exactly what the blue stain is in the middle. But you can certainly imagine then that if the capillary is already tight to begin with, now the cancer cell is in this mass. Okay. And also think about the fact that the blood is pushing these cells through it, right? So you have this bulky mass that's being shoved through this teeny tiny capillary, and the actual flow itself can be a lot rougher than a typical cancer cell is expecting, since it just came from nice, quiet tissue to start with. So it's very much like riding the rapids in a raft. It's a rough environment, right? And if cells are not equipped to handle it, they can really be destroyed or torn apart. Okay, let's move to the second part of our lecture. So the cells have now entered the blood. They are associating with things like platelets, etc. Eventually, right, they're going to end up going into the veins, making it back to places like the heart, and then the heart's going to pump them to places like the lungs. And then when that blood comes back, the heart's going to pump it out to different places in your body. your brain, places all kinds of tissues, including bone. Right? So let's think about what's happening then as these clumps of cancer cells and platelets are being shoved throughout your body through the circulation. So once they're in circulation, we call them CTCs, or circulating tumor cells. Within just a few minutes, circulating tumor cells that have entered the blood from any part of your body will have made it to the heart and then are pumped to the lungs, right? Because that's how our circulation works. In the lungs, they're going to get stuck in capillaries, especially because they're these big clumpy platelets. And once they're stuck in there, it's almost like a clogged drain. There's going to be a buildup of pressure behind them, and that might actually end up shearing off pieces of cells. Right? One of the strange, bizarre properties of cancer cells is that cancer cells can actually lose nearly all of their cytoplasm and still survive, again, because they're sort of all messed up. Now, I'm not saying that most of them would survive, but it's just another selective event that happens to select for those cells that can survive. So if they're stuck in a capillary in a lung and their cytoplasm gets shredded off, that might make them small enough to then dislodge from the lung and actually make it back to the heart to be pumped someplace else in the body. So here, let's take a look. So here we have a cancer cell going through the blood, right? It's associating with different platelets. The platelets begin to form a clot, right? Which actually makes it even harder for this cancer cell to move through the blood. So it's really getting lodged here, and this clot is forming around it. So as a result, the circulating tumor cell gets caught in a capillary somewhere else in the body. And if you think about it, now it's very far away physically from that original reactive stroma environment that it had developed in, right? The reactive stroma back in its original home that ultimately ended up kind of chasing it from its home. If we go back to that dysfunctional teenager analogy, right now the teenager has wandered away from home, gone somewhere far from home, right? And because they're away from that bad emotional environment of the home, maybe they sort of settle down and chill out and suddenly began to regain their senses and realize, right, that they really wish they were home again, that they're scared, they don't know where they are. So the cancer cells are like that too. Once they're away from the reactive stroma environment of the original primary tumor cell and they find themselves stuck somewhere else in normal tissue in your body, right, they kind of get homesick. So again, as part of the wound healing response, you can imagine that we evolved to not want to have clots of platelets floating through our blood, right? That's really bad. That can cause all kinds of problems in terms of clogging up important blood vessels. I think you all know that when you have things like thrombosis, this idea of people, you hear stories about when they sit on an airplane for a long flight, that somehow they can develop little clots in their blood vessels in their legs. And if one of those clots ends up getting lodged loose, it can go up and actually get stuck in somewhere, like in their heart muscle or something like that, right? So not surprisingly, our body then doesn't like clots floating through the blood. So we actually have anti-clotting mechanisms to resolve or dissolve clots. Okay. So here we have the clot forming. The clot has formed. That enables the cancer cell to get stuck in place. And importantly, at this point, the platelets are ironically protecting the cancer cell from detection by things like immune cells. But in this environment, the cancer cell is now quote unquote homesick because it's away from those confusing dysfunctional messages of its primary tumor site. And so now it wants to become epithelial again, right? So it needed to go back at its original tumor from epithelial to mesenchymal properties. So that was the whole EMT, or epithelial mesenchymal transition, that enabled it to go through the stroma and to the blood. But now that it's stuck somewhere else, far from home, it wants to revert back to becoming epithelial. And one of the things it wants to do, right, one of the things that it loves to see is a basement membrane. And so it begins to want to attach to a basement membrane. And in this case, it's the basement membrane that surrounds the capillary. So this microthrombus or microclot in the blood, right, provides an opportunity then for this cancer cell to stop, seek out the nearest basement membrane, and it adheres to it by shoving aside the green endothelial cells in the picture here. Okay. So this brings us to our next step then, which is the tumor cell stuck to the side of a capillary and wanting to exit the capillary. So we call this extravasation, or exiting the vascular system. Okay. And they basically elbow their way out between the endothelial cells because they are seeking the basement membrane. And once they are in the stroma, right, they are now back in a healthy, normal stroma environment that just further encourages the cancer cells to want to resume their normal epithelial behavior. Right? And that includes dividing. At this point, they will start dividing and form just a few cells, a microtumor. So let's look at this extravasation step. So these panels on the top, we looked at in a previous slide. Right? This is when the cancer cell is now seeking the basement membrane and wants to leave. I mentioned that the microthrombus or the microclot actually will be dissolved by normal pathways or normal functions of our body that doesn't like to have clots floating around. So the platelets will be shed, and that then enables the cancer cell to really begin to want to seek out the basement membrane. So here it's kind of dividing now, because it's receiving maybe growth signals from the normal stroma, this gray region on top of it signaling through the basement membrane. They begin to divide, and they really want to be able to establish themselves in a new home, right? In a normal stromal environment that they're very homesick for. And this then encourages them to migrate into the surrounding stroma or the surrounding parenchyma. Remember, I told you parenchyma is another term for the surrounding tissue around things like blood vessels inside organs. So here you have a great set of illustrations that show this. So here is, in dark brown, this big thick thing is a small arteriole that has entered this tissue here. You know, this is the normal blood supply. And you can see that throughout along the way, the arteriole is branching, right? Branching into small, tiny little capillaries that grow off of it, right? And here's a big branch and you have smaller capillaries. So stained in fluorescent green here are the circulating tumor cells, or CTCs, that just by chance, because remember, it's kind of strange to think about it, but as they're flowing through the blood, every time the blood vessel that they're in makes a left or a right, like a fork in the road, you know, who knows which way they go. Maybe one way they go left, one way they go right, left, right, whatever, until they eventually end up in a situation like this, where it's the end of the arteriole, and now they are kind of lodged in these teeny tiny capillaries. But you can see that some of them represented here are moving out of the capillary into the surrounding tissue. So this ability to leave the blood supply certainly is not something, you know, new that's invented by the cancer cell, because we have many cells in our body that do that. For example, our immune cells that regularly go around in our circulation until they reach a part of tissue where they need to leave, right? And when something like a white blood cell leaves the capillary and goes into the surrounding tissue, and becomes something like a macrophage or something like that, we call that process diapedesis, the exiting of a cell from the blood supply into surrounding tissue. So leukocytes, white blood cells, like monocytes that are going to become macrophages, they're obviously programmed to do this. So it only takes 1 to 2 minutes for diapedesis to happen for a leukocyte. On the other hand, you have to imagine that the circulating tumor cells, right, they are not programmed to do diapedesis. They don't know how to do it. What are the chances that they actually know how to do this? Right? So that's why for them to escape the blood, it takes hours or days. But once they are in their new surrounding tissue here, we no longer call them CTCs, right, because they're no longer circulating. We now say they have been disseminated or spread out now to new destinations throughout the body. And so we call them disseminated tumor cells, or DTCs. Once they're in their new stromal environment, remember, this is going to be totally normal, healthy stroma, right? They are going to maybe divide a few times and form one of these micro metastases. So here you have colon carcinoma cells in red that by chance made their way to bone. Okay, so now they're inside the bone marrow because they hitched a ride through the arteries that eventually got smaller and smaller arteries into a bone and became little capillaries. And then these colon cancer cells that were CTCs, as they're circulating, now underwent diapedesis and are now in the bone marrow. And you can see they only divide a few times. Right? Here you have breast cancer that metastasized to bone. Same thing, you have a micro metastases that may be no bigger than just a handful of cells. And surprisingly, they may stay this way indefinitely. Right? Why? Because this new stroma that they're in, right, even though it's healthy, normal stroma, it's not the stroma that these original cells knew how to live in. Right? It's not the stroma that's sending the messages that, for example, colon cells used to get when they were in the intestines, or breast carcinoma cells used to get when they were back in breast tissue. So these DTCs, these microtumors, may lay dormant in these metastatic sites forever. And if you think about it then, when you think about how many of these circulating tumor cells are being released into the blood by a big tumor, remember earlier in the lecture we saw a wonderful image of these pink tumor cells, and throughout were all these little dots of where the capillaries were going through that cross-section of the tumor. And I asked you to think about how each one of those little cross-section capillaries, there are several tumor cells shoved up against it. And so if they're all making their way into those capillaries, right, and that's happening every moment throughout the entire tumor, we're talking about hundreds and hundreds of thousands of these tumor cells getting into the blood every day from a site like that, right? So you have hundreds and hundreds of thousands, if not millions, of circulating tumor cells going through the person's body, right? And that means you're going to have thousands upon thousands of them getting lodged in random places throughout the different tissues and exiting and becoming little micrometastases. So that means that by the time a patient presents in the oncology clinic with symptoms due to a tumor, so pain or blockage, even things like a pinched nerve as a result of the tumor, by then, even though on the initial x-ray, they might say, oh, we identified this main tumor here, this is the primary tumor, by then, there may be thousands of micrometastatic tumors scattered out throughout the person's body. Right? So by the time the primary tumor is treated, it may already be that that person has thousands of other potential little tumor sites already forming. But as I said before, these very well might be non-growing DTCs. Right? And one of the problems with this is if they are not actively dividing, then these little metastatic cells may not be responsive or sensitive to drugs that are designed to target actively proliferating cells. So many of the first line chemotherapies that are given to a patient who have a tumor, maybe the tumor is in a place that cannot be surgically dealt with because it's too risky, or maybe even if they're going to perform surgery, as part of the pre-op, in the weeks leading up to it, they give the patient anti-tumor drugs to try to shrink the tumor to make it easier when they finally do undergo surgery, right? Many of those drugs are meant to preferentially target cells that are actively dividing, right? Which means normally you think the tumor cells. But what about all these thousands of teeny tiny micrometastases where these metastatic cells ended up in some stroma somewhere else in the body? It's not really the right kind of stroma that they know what to do with. And so they maybe divide a few times and then just stop, right? So those drugs might not be able to be effective against those. Okay, where's my mouse here? oops, it seems like okay, sorry, my slides seem to have crashed here. Let me stop the recording for a moment and see if I can, oh no, the whole thing's crashed. Okay, there's a couple more slides. We still have time, so we can go through with them, but it looks like I have to stop the meeting and start it up again. I apologize for that. So why don't we log off right now, and right away I'll close this, reboot, and start the meeting again. Okay? Okay. Okay. It looks like we're back here. Okay, sorry about that. So let's talk about then, what is it that prevents these micrometastases from actually being able to kind of start growing into a new, new tumor in the site of wherever it was that they've disseminated. Okay, so that takes us to step 7 then. Right? In order for these micrometastases to really truly colonize and begin to form another macro tumor or secondary tumor, an important thing to keep in mind is that these DTCs, these disseminated tumor cells, must be happy with whatever new stromal tissue they've ended up in. And as a result, the vast majority of disseminated tumor cells won't end up being able to successfully start growing in their new tissue. They just divide a few times and they stop, and they just stay there as a micrometastases. And part of this is they need to be able to properly make sense of this new environment that they're in, and make use of signals that they know how to respond to enable them to successfully revert back fully into their epithelial growth and start growing. So this is a very important concept here. And this is one of the, if you study cancer, there's this very famous kind of concept known as the seed and soil model of metastatic cancer. And it basically likens the disseminating tumor cell to a seed for a flower. If I were to give each of you a handful of wildflower seeds, and you were to wander around someplace like the UH Manoa campus, just kind of randomly scattering seeds in different places, like maybe you scatter a few of them in a nice sunny grassy area, maybe you scatter a few of them in some dirt by a parking lot, maybe you scatter a few of them right on the sidewalk, right? And then you were to go back weeks later, you certainly would not be surprised to find that certain places where you scattered these seeds on the campus now have these little wildflowers that are growing. And you would not be surprised that other places where you scattered the seeds, there's nothing, right? Because the environment that you put the seeds in are just not the right environment that's compatible for them to grow in. So if you think about the different types of tissues in our body as being the potential new stromal environments for these disseminated cancer cell seeds that are scattered around, likewise, right? Only the cells that ended up in compatible stroma or compatible soil will actually flourish and begin to grow. So therefore, the emergence of health-threatening macro tumor metastases throughout the body is actually a very rare subset of the disseminated tumor cells that are being spread all around. Okay. So here's a nice slide that kind of puts together the concept then of epithelial mesenchymal transition and the cells having to revert back to being epithelial. So here we have back at the primary tumor site a large tumor mass that's made of many different subpopulations of tumor cells. So recall, all of these, if you trace them back far enough, years and years and years ago, started from one cell. But as the the mutations began to pile up in these cells, some of them stumbled upon a particular mutation as their next mutation. So they would be like the orange ones here. Some of them maybe didn't do much. Maybe they are the original gray ones, for example. And so then you have different populations of cells with different potential, right, to be able to actually make the epithelial to mesenchymal transition that leads them to becoming disseminated. Okay? So let's say all of these cells here, okay, make it in terms of being able to be shed into the blood, and they begin to go around in the blood, right? So they are disseminated and they form micrometastases scattered throughout the body, right? But only some of them really kind of score by ending up by chance in tissues that provide them the signals. In other words, the right soil that the seeds have ended up in to allow them to become macroscopic metastases, right? And a big part of that then is them undoing the epithelial to mesenchymal transition and reversing the process, becoming what we call mesenchymal to epithelial transition, so that they resume their epithelial properties. Right. So this concept of reversibility of EMT, right, in other words, MET, right, is one of the features of metastases. So here we have on the left the image of the original primary tumor with the basement membrane in green, right, and then the cancer cells or the tumor cells in red. So they undergo EMT and begin to break free of the basement membrane, right? They break free of the basement membrane is deteriorating, and so now these cells are released into the blood. They eventually end up somewhere else. So wherever this primary tumor was, some of these circulating tumor cells end up being disseminated tumor cells in some place like the liver, right? And they revert back to their epithelial state, and you can see they sort of happily resume their growth as though they are the primary tumor with a restored basement membrane, right? So those relatively rare micrometastases have really lucked out by growing in good soil and resume their epithelial-like growth, right? And keep in mind then, I put a little asterisk because this is very relative when I say relatively rare, but you have thousands of micrometastatic sites. So even if you say, oh, it's very rare, only a handful of them, but that's still a problem if you have a handful of sites in the body where the secondary metastatic tumors can in fact grow. So this cartoon then just summarizes then the reversibility of EMT, right? So we don't have to go over this together because I think this is sort of a summary slide that really puts into a nice cartoon form the concepts that we talked about today. So I encourage you as part of your review to talk your way through this slide and think about the different events that are happening that we discussed.